Good afternoon. Welcome to this year's Marshall-Nirenberg Lecture. This lecture was established only a few years ago in 2011 as part of the Wednesday afternoon lecture series to honor Marshall's work in deciphering the genetic code, which resulted, of course, in his receiving the 1968 Nobel Prize in Medicine or Physiology. Marshall's research career at the NIH spanned more than 50 years, and towards the end of his career, he began to work in neuroscience, neural development, and homeobox genes. He was an active member of the NIH intramural community until his death at age 82 in 2010. And so the lecture was established to commemorate his time at the NIH and his amazing achievement. The lecture that bears his name recognizes outstanding contributions to genetics and molecular biology. This year also marks the 50th anniversary of Marshall and his colleagues at the NIH fully deciphering the genetic code. That effort started with synthetic RNA and milieu, you all know those experiments, in the early 1960s, and culminated with a series of papers delineating the entire code in 1965. I make this point because there's an argument about when to celebrate the 50th anniversary, so we decided to wait until the code was completed. So we're having a little fun this year with the Nuremberg lectures. This is actually the third Nuremberg lecture, recognizing the fact that the code is a triplet. And so we're having 3 lectures to honor Marshall. In March, there was a ceremony at the National Library of Medicine with speakers remembering Marshall's career. We also recognized the generous gift to the NLM of Marshall's Nobel Prize medal and certificate by Marshall's widow, Myrna Weissman. They're now on display at the NLM Building 38 on the NIH campus here, and if you get a chance, you should go over and see them. On May 20th, we had the second Nuremberg lecture, in which David Page presented a talk entitled Lost in Translation, Do Males and Females Read Their Genomes Differently? Very timely because of the interest in gender differences in biology. And if you missed it, that lecture is, of course, archived, and you can get to it by going to the NIH website. And today, our third event, and I won't say anything about this being a wobble event. Hopefully it will be a steadfast event. We're fortunate to have Tim Ley as the Nuremberg lecturer. By the way, a nice exhibit of Marshall's work is on display outside of the Lipset Amphitheater, and I encourage you to see that if you haven't already. Tim Ley is the Lewis T. and Rosalind B. Apple Professor of Oncology in the Department of Medicine at Washington University School of Medicine in St. Louis. He's a hematologist, oncologist, and cancer biologist. Tim is internationally known as an expert in the pathogenesis of acute myeloid leukemia, or AML, and other blood-related disorders. His research group was one of the first to focus on the reference sequence of the human genome to systematically identify the mutations responsible for the initiation of progression of AML. And in the instance, particular first instance, this led to improvements in the treatment of this disease. To better understand the role of these mutations, Tim and his colleagues have constructed mouse models of acute promyelocytic leukemia, which have many features of the disease in humans and have turned out to be extremely helpful. And I don't know whether Tim will discuss those today, but if not, you should look up his papers on that. Tim's lab has long been interested in the tools and methods needed to correct single gene defects. They're using both genetic approaches and expression profiling to identify the homologous recombination signature to improve the efficiency of this process in both embryonic and hematopoietic stem cells. Tim received his BA from Drake University in '74 and his medical degree from the Washington University School of Medicine. He did his internship and residency in medicine at the Massachusetts General Hospital, and he was both a clinical associate and then a senior investigator in NHLBI at the NIH before moving to Washington University. He's a member of the National Academy of Medicine and the winner of numerous awards, most recently the 2015 Erasmus Hematology Award. He served for years as the chair of the BSc for NHGRI, and this year, President Obama appointed him to the National Cancer Advisory Board. So without further ado, let me introduce Tim, who will speak on the AML genome mutations in 4 dimensions.
Thanks, sir. Appreciate it. Good job. Thanks for the invitation to do this lecture. I'm truly honored. I didn't know Marshall, but I'm here because of him. We all owe him a great debt. He was a hero to science. This is the man we're honoring today. Note in this picture of him, he has a dirty white lab coat on. He's wearing a tie. Pay attention to these details. This is after the code was broken in the early 60s. I came here in '79. I had an interview with this man looking at a clinical associate position. This is Jack Orloff, who was the intramural director of NHLBI. And my background in research was minimal. I done nothing in medical school. Most of the other candidates for the clinical associate post had a PhD or done a lot of research. And so I was pretty concerned that I wasn't going to get a position. So Jack, in his inimitable way, asked me if I'd done any other research at any other time in my life. And I said, I did do some science as an undergrad. And he goes, what was it? And I said, well, we studied the distribution of macroinvertebrates, benthic macroinvertebrates on the Des Moines River, above and below the city and above and below dams, and just collected bugs to see whether their distributions were changed by damming this river. He was unimpressed. He said, what kind of bugs? And I said, caddisflies, predominantly, and dipterans. And he said, oh, I'm not impressed. And he said, how many did you count, classify? I said, well, hundreds of thousands. He said, okay. And he was ready to dismiss me, so I threw my career Hail Mary. I said, but you have a famous scientist in your own branch in the Institute who's a caddisfly expert. He said, really? I said, yes, you do. He said, who? I said, Marshall Nuremberg. He said, bullshit. So he got up from my interview ready to dismiss me from the room, but he decided to go into his office and call Marshall. Came back a minute later, and he said, well, Marshall says that counting and classifying caddisflies is obviously very good training for a molecular biologist. So you're hired, kid. So that's how I got here. So here's a picture of a caddisfly that he drew, a camera lucida drawing that was in his master's thesis about caddisflies and water quality. Even at this early stage in his career, he was prescient. He wrote in his master's thesis that the fundamental importance of these caddisflies has been overlooked, and they appear to be highly sensitive indicators of stream pollution. So the exact idea that we used 20 years later to integrate all the pollutants in the Des Moines River. It's funny in other ways. The funniest comment in his master's thesis is this one. Welcome to science, Dr. Nuremberg. In general, for every problem solved, 2 problems were raised, which are still unanswered. He was adventurous in his early days. Here's outside a swamp in Florida where he obviously doesn't look very perturbed at all to be holding a 7-foot-long snake. And 50 years ago, as Michael mentioned, with Phil Leader and others, he solved the genetic code. This is the paper in 1965. So as most of you know, this was solved on the multiplater, the Leader multiplater. And there's a version of this in the NIH Museum. What most of you don't know is that there's another multiplater out there. And here's David Ressler Germain, Ron and Gail's son, working on the other multiplater in my lab, which was given to me when I left the NIH by Art Nienhuis because nobody else was using it. So it's been in continuous use for the past 30 years in my lab, and some of the data that David generated from this will be shown later on. So there he is, and he's defending his thesis in 3 weeks, and look forward to having Ron and Gail there. So he's touched my life in many ways. I'm here because of him, and we're here today to honor him. So let's talk about AML. For those of you who don't think about this disease all the time, I'm going to talk for a minute or two about its clinical features because in modeling the pathogenesis of this disease with genomics, it's critically important to pay attention to all these clinical features because any model that you develop clearly has to explain the clinical presentation of the disease. So this is a very heterogeneous disease, almost a syndrome, lots of different morphologies, cytogenetic findings, and clinical features. About 19,000 new cases per year in the United States, but 14,000 deaths, unfortunately, and that hasn't changed much. Often the presentations are explosive. We'll talk a little bit about why that is later on. The average age of presentation right now is 67 in the United States with a 5-year survival of only 5%. Pretty depressing. And death usually occurs within weeks to months unless you can establish a first remission. MDS, myelodysplastic syndromes, and AML are diseases of aging. They definitely have an enormous age distribution that really starts to pick up in the 50s and 60s. And one other important feature of the disease that you have to understand to model it is that the incidence of this disease is constant, and it's constant all over the world. And this SEER data definitely suggests that it must be the product of a group of fixed variables. And that's also important in building a model for the disease. We treat the disease pretty much the same way that we did when I was a fellow 30 years ago. Clearly one of the great disappointments of a career, not to have made big changes in therapeutics. We still give people 7 and 3, 3 days of daunorubicin, 7 days of cytarabine. And after they go into remission, if they do, the 80% that do, if they're thought to have good to intermediate risk AML by cytogenetics, we give them high-dose Ara-C for consolidation, or we transplant them if they have high-risk disease. In older patients, refractory, relapsed patients, secondary AML, treatment-related AML, standard of care is a clinical trial. We don't know what to do for these patients, and if there is no clinical trial, we make up treatment protocols based on best knowledge and experience. And also, we give inhibitors of DNMT1. Don't know how this works. I actually did the first clinical trial in AML at WashU when I was a fellow there in 1983. Because the response rate was 25%, my mentor thought it was noise and said, forget it. So DNMT1 inhibitors are thought to be palliative, can sometimes bridge the transplant, but they don't cure the disease. So for many years, we've used cytogenetics to classify patients as they present. When they walk in the door, we do cytogenetics, and they have one of 3 kinds of disease. Favorable risk, one of 3 translocations that are canonical. These patients can be treated lightly, and they tend to do better. Unfavorable risk, these are usually people with aneuploid disease, complex cytogenetics, almost always with P53 mutations underlying this disease. And unless these patients get transplanted in first remission, they do very poorly. And unfortunately, though, cytogenetics does not classify this group, intermediate risk, most of which have normal karyotype disease. So the big question when it became possible to think about sequencing these genomes was whether by defining all the mutations in this disease we could improve risk classification for intermediate risk patients. So after figuring out how to sequence these genomes and do the analysis in a fairly rapid fashion, we selected 200 cases of AML that kind of represented the whole gamut of disease that we see. It would look like 200 patients that came through the door consecutively. The only thing that went into the selection of these cases was sample availability and the ability to model all of the classical cytogenetic subgroups of the disease and morphologic subgroups. So we sequenced 50 of these with whole genomes and 150 with exomes. This was published a couple of years ago in the New England Journal. And a couple of big lessons. One is these are minimally mutated genomes. There are only an average of about 13 mutations in the exome per case, which is very, very low, the lowest of all adult tumors. And only 5 on average are in recurrently mutated genes, about 1,000 mutations across the genome in each case. It's much, much lower than other kinds of solid tumors and other malignancies in humans. We found 23 significantly mutated genes when corrected for gene size. The big 3 are FLT3 and nucleophosmin and DNMT3A. And these are together because they tend to co-occur. And they cooperate in some way that we don't understand yet, but they're very important. And I'll show you the order of events in a minute. But the other genes tail off fairly quickly to the 260 genes that are recurrently mutated. And the 1,600 or so singletons we found in this cohort, some of which clearly fall into classification schemes like the one shown here. So we put this group of 9 kinds of mutations together. Everybody thinks we did this with some fancy algorithm, but it was just kind of me sitting down and going, this looks good. Anyway, there are 9 groups, and the mutations cluster in these groups to some extent. And there are rules of mutational exclusivity and cooperation that are apparent, that are very important for understanding the biology of the disease. But the important thing is we scaled this exercise so that we could find almost all of the mutations that occur in at least 5% of AML cases, so that we could use this information, along with many other groups around the world, to improve risk classification. So as soon as this paper came out, the large groups that had done lots of sequencing got to work and tried to incorporate all these mutations in panel tests with people who had known outcomes. And even though the expectation was that this exercise would improve classification, it did not. So it had not solved risk classification. So this was a major bump in the road for us. We thought this would work, but it clearly didn't. So the question really is why? Well, the first thing that occurred to us was that we missed mutations in the non-coding part of the genome, because only 50 of the cases had been sequenced throughout the genome. So we simply asked whether we missed mutations by sequencing exomes. So we took a little different approach. We didn't take an all-comers approach to test this idea. We picked cases that either had done well for AML with a long first remission with chemotherapy only, or we took cases that had primary refractory disease, never entered a remission, or that relapsed right away, and we sequenced their genomes. So here's the whole genome view of these mutations. So here's chromosomes 1 through X and Y down here. And each one of these is the number of times we encountered this mutation in these 71 genomes. So down along the bottom here are all the non-coding mutations in 10 kilobase windows. So a whole bunch of them are recurrent. But they're kind of recurrent at the level of noise. Some of them probably are recurrently mutated regions that might have some function, but you can see that the maximum number of cases that had these non-genic mutations was only 2 or 3. So none of them were risk classifiers. And these are all the recurrently mutated regions that contain the canonical genes that I just showed you. So there was nothing in the non-coding space that was useful in terms of classifying risk. So we didn't miss it by sequencing exomes, as it turns out. So here's the summary on this story, which was published a few weeks ago in JAMA. So whole genome sequencing of short versus long remission AML. So one gene was a classifier of good risk, nucleophosphate mutations, which we already knew. A few genes were associated with early relapse. These were just univariate trends, not highly significant. They'll fall apart when more cases are done. As I told you, the recurrent non-coding variants, there were none associated with a risk. There were no germline mutations associated with risk. There were no associations with numbers of mutations in these cases in risk or numbers of subclones. And there were no epigenetic signatures that were predictive of risk. So what went wrong here? Well, it's a complicated disease. And I think that the way we think about it now is that there are a lot of confounding variables. And there are really a number of dimensions of this disease which have to be taken into account if you're going to understand why risk assessment at presentation doesn't work. So first of all, there's just mutational complexity. Each patient seems to have 3 or 4 mutations in important pathogenetically relevant driver genes. And when you multiply that over several hundred genes that can be mutated that are relevant, you get an enormous matrix. It's almost infinite in the number of combinations that you can see, just at the level of saying genomes have a mutation or they don't. Simple, binary, one-dimensional genomics. That matrix is absolutely enormous, and it may be so enormous that no 2 AML patients will ever be exactly the same. The second thing, though, is clonal complexity, which we bumped into and understood right away because of digital genomics. And basically, all AML genomes that we've sequenced to date, now several hundred, are clonally complex. There are probably no exceptions to this rule. So every one of them consists of a founding clone and 1 or more subclones derived from it, usually multiple. And that number isn't really known for most of our cases because we really haven't done a good job in covering these cases deeply enough to know how many subclones there are. I'll show you that in just a minute. So all the sequencing led to the development of this simple model for the pathogenesis of the disease and the origin of the mutations. So one of the things we figured out fairly quickly was that there is a mutation rate in hematopoietic stem progenitor cells of about 1 mutation per cell division. Now, these cells are long-lived. They live for decades. The group that you're born with, you die with copies of those same cells. But every time they divide, they pick up one mutation. They divide about once a month. You have about 10,000 of them. So by the time you're 50 years old, every single one of your 10,000 hematopoietic stem cells has a unique genetic signature that includes about 500 mutations. You have a 1 in 2 chance of having a functional P53 mutation in one of your hematopoietic stem cells when you're 50. 50% chance. Doesn't matter, though, because unless it sees a stressor, that cell has no competitive advantage and does not contribute to abnormal hematopoiesis. So the idea is that once in a while, one mutation occurs that gives the cell a very, very small advantage. These are not earthquake mutations. These are just little nudges, very soft mutations, so that there's a pre-leukemic advantage of cells containing this mutation. This was a little controversial at first, but I think last year the discovery of the same group of mutations in elderly people with clonal hematopoiesis really anchored this idea firmly in all of our minds. So if you just look back at TCGA data or other large cohorts as other groups have done, you can see that mutations start to skew hematopoiesis in elderly people, so that by the time you're in your 8th decade, at least 5%, absolute minimum number, this is just low coverage exome data, at least 5% have mutations that are skewing hematopoiesis towards a single clone. And the mutations that they have are the same ones that are associated with the initiation of AML and MDS, DNMT3A, SXL1, JAK, SF3B1 is splicing mutations in TET2. So this idea that there is this skewed clonal hematopoiesis with normal blood counts in the elderly has really helped this hypothesis to go forward. It's now called CHIP, clonal hematopoiesis of indeterminate prognosis. About 4% of people with this go on to develop MDS or AML. We don't know why those 4% do and 96% don't, but it's a risk factor for developing these diseases. So nothing happens until you get another event, a cooperating event, which then can form a founding clone, which really is AML. And one of the properties of this founding clone is that it loves to experiment with additional mutations that give its progeny, its subclones, an advantage. And so these subclones, of which there are many in each individual patient most of the time, clearly have an advantage and outgrow their parent. The reason that you can detect them with the sequencing technologies that we use is because they have a growth advantage. Even if a subclone is only 1 or 2% of the cells, it's growing faster than its parent because you can detect it. So the usual order of events in the most common form of normal karyotype AML is a DNMT3A mutation, which is the initiating hit. It's cooperated with a nucleophosmin mutation, and then it explodes with a subclone that acquires an activating kinase mutation, like an activated FLT3 mutation. And we think this is why the presentation of this disease is so explosive in many patients. It's that final hit. The cells have somehow been inured to a strong proliferative signal by the upstream mutations. They can tolerate a massive proliferative hit, and the disease can literally explode. When we sequence the patient's genomes and perform a plot of each mutation with the varying allele fraction versus coverage over here, you can see these mutations cluster in groups. So this, in fact, is a big cluster of mutations that represents probably 1 or 2 events that contributed to the founding clone and 499 that it dragged along in the stem cell that was transformed. And then there's another cluster here, which is a subclone. And how do we know this? Because early on we sequenced individual cells from these patients and proved that these subclonal events contained all the mutations that were in the founding clone, plus a new set, usually one of which is relevant in the subclone. So this is what these kernel density plots look like when we sequence these genomes. There's a founding clone that is the largest group of cells, and subclones which have evolved from it, which frequently are the dominant cell at presentation, but they don't represent the founding clone per se. So when we've looked at now hundreds of cases, these are the kinds of patterns that we've seen. Every one of these cases has got clonal heterogeneity. Here's one that kind of looks to be a single clone, but this is with 30x coverage of the genome to discover all the mutations. And in fact, it has subclones that I'm not showing you here. Here's one that we just looked at with founding clone, one subclone, Here's 2 clear subclones. Here's 3, on and on. But this is all based on initial sequencing of 30x coverage or 100x coverage of the exome. And it really does not do justice to the subclones that are sitting down at the bottom, which you simply don't cover well enough to detect. So we've looked at how much we're missing. A lot of people think this can be solved by just going deep on a small set of genes, panel testing, if you will. So here's a case, AML 31, where we use the panel test consisting of all the known recurrently mutated genes in AML. And this is quite a heavily mutated AML case. So you can see a few mutations that are probably in the founding clone and a few that define subclones. But it's not enough to really understand the clonal complexity of this case. So here's the same case sequenced with an exome reagent, about 100x exome. So it looks more like a founding clone in the subclones. And here's the same case, which we just published, sequenced to 300x for the whole genome and then validated. This is kind of an expensive experiment. Well, this is 30x, and here's 300. So there's 30x, what I've been showing you, where it looks like there's 2 subclones. And then if you fast forward this to the 300x exome, you can see what we missed. See everything we missed? So most of it's down at the bottom. So why do we care about that stuff down at the bottom? Remember what I said. The stuff at the bottom you can detect because it's growing faster than the parent. So let's look at this genome a little bit more and ask why these subclones matter. Why are we so concerned about them? Well, for one thing, as we think about targeted therapies for these diseases, it's really critically important to think about where the targeted mutations lie in the clonal hierarchy of the disease. So look at this case. The founding mutations are DNMT3A and nucleophosmin. This is a subclone that contains an IDH1 mutation, for now which there's a targeted drug, FLT3 D835 mutation, and it also has a FLT3 ITD mutation. So in the old days, we used to think these were biallelic mutations. Now it's clear that these kinds of mutations frequently occur in completely different cells, and there are 2 different subclones that have been growing because they're being pushed by these activating kinase mutations. So if you gave a perfect IDH1 inhibitor to this patient, what would happen? This subclone would be cleared, and other subclones would replace it. If you gave a great FLT3 inhibitor to this patient, it was 100% effective. You would clear these 2 subclones probably, and you would be left with the founding clone, which would reshuffle the deck, and you'd be right back where you started. So that comes to the topic of clonal evolution with time, the 3rd dimension of genomics and AML. Let's look at this same patient's genome that's fully resolved now with a 300x genome at the time of presentation and at the time of relapse. So here's the same case. So I have the varying allele frequencies presented down here at presentation, and here they are at relapse. So at relapse, the founding clone reappears with all of its mutations, DNMT3 and nucleophosmin. With standard induction therapy and high-dose RSC consolidation, this patient cleared 3 different subclones, 2, 4, and 6, the ones I just showed you. So that's what these completely were resolved by standard therapy. And the mutation that comes back at relapse is this one and this one. So this one contains a different mutation in IDH2. It was sitting in the weeds, and it was selected for with chemotherapy. And here's another clone down here that may or may not be a part of the initial event that led to this AML that contains a P53 mutation, which was strongly selected for by chemotherapy. So this just begins to show you, when you really get to depth and understand these genomes completely, how unbelievably complex they are, and that these subclones appear to have different rules in terms of how they respond to induction therapy. So this is actually what this case looks like when you put it all together over time. So there's the founding clone in gray. 3 different subclones appear at presentation. 2 are eliminated, and a tiny one, which you're not seeing here. 1 goes forward. A mutation within it expands after consolidation therapy and then blows at the end. And this P53 clone, which was in a different stem cell, also expands with induction and is present but probably not contributing to relapse per se. It's complicated. And the other thing that complicates things is that these subclones clearly have different functional and morphologic properties. They're very different. In a study that Jeff Clow and Dave Spencer did, it's clear that some of them have unique immunophenotypes, some don't. Some of them have altered growth properties when you plate them in culture. There's tremendous engraftment bias in xenotransplantation models, so that if you put in an AML with 4 subclones, it's highly likely that in each individual mouse, only one will engraft. And quite frequently, only one of the subclones is capable of engrafting. And recently, as we've made IPS lines from AML samples, usually only one of the subclones is fit for reprogramming. Some of these subclones contribute to relapse and some don't. And unfortunately, right now in the early days, it's not at all clear what the genetic rules are for the behavior of these subclones. So we can't just stop. The question really then became, how do we incorporate this information into understanding how to classify patients? We somehow have to integrate this clonal heterogeneity in response to induction therapy in the individual to understand better how to move forward. So we asked this simple question: how do we integrate this information into risk assessment? Simple idea: the subclones respond to induction therapy differently, the different rates of clearance. So a very simple experiment that we completed and published a few weeks ago in JAMA. We just took a bunch of cases, 50, and evaluated the clearance of all the leukemia-associated variants that were present on day 0 when the patient recovered from induction therapy on day 30. That's about when we get the second biopsy. All the cases that we examined were in morphologic remission. No point in looking at people who weren't. So these are people who, based on current standard of care testing, have all done well. They're in remission on day 30. And let's just see, when we sequence them to a sensitivity depth of 1 cell in 100, how many of them have really cleared. So there are 3 kind of basic patterns of clearance that we saw. One is good. Here's the blasts here, which go to 0. They're 75%, they go to 0. This patient clears all the mutations to a threshold of less than 1%. So every mutation basically goes to 0. And patients like this generally have gone on to have long first remissions with chemotherapy only. Here's the second pattern. All the variants persist on day 30. Not good, as you would expect. Remarkably, though, this patient had normal-looking hematopoiesis on day 30. Just shows you how insensitive that test really is and how much we've been missing by focusing on morphology. You can have differentiated cells that are full of leukemic mutations. So this didn't surprise us. This is the one that kind of caught us by surprise. This notion that in this patient who clears his blasts completely, down to nothing, has this response because he clears a subclone completely. So the dominant cell in this patient's marrow was a subclone, and that clone was completely cleared, had an activating RAS mutation, and so probably very sensitive to this kind of chemotherapy. And the DNMT3A and IDH1 mutations, which were part of the founding clone, just laughed at this chemotherapy. I mean, absolutely untouched with ablative chemotherapy. Imagine. And then persist, and what I'm not showing you here is that new mutations appeared in a subclone of this, reshuffled the deck, that contributed to relapse. So it's these 2 mutations plus more. So when we look across all 50 patients at how they cleared their mutations, there are 2 patterns. One group of 26 patients cleared everything down to a threshold of 1%. And these patients had an event-free survival of about 18 months. And many of them in white are still in remission. But this group here, if you clear, you're blue. If you have a persistent mutation, you're in red. Even the persistence of a single mutation in this group on day 30 is bad. The event-free survival for this group of people is 6 months. And what is kind of interesting is that what are the mutations that they fail to clear? And they tend to be these initiating events. These are the ones that are tough to get rid of, the ones that are in the founding clone. And so ones in DNMT3A and TAT2 and IDH1 and 2, if they're in the founding clone, they are the ones that tend to be persistent after successful induction therapy. And these ones that are contributing by cooperating or by providing a big proliferative signal are the ones that are generally cleared, suggesting that those subclones are somehow more sensitive to the kinds of induction therapy that we're giving, which kind of makes sense, I think. So anyway, this matters. Event-free survival, it's shown in Kaplan-Meier. If you clear all the mutations, you're here. If you don't clear them all, even one, you're here. And overall survival is also affected. And importantly, this holds up very nicely for the 32 patients who had intermediate risk. So now we can begin to think about classifying them more accurately as well, since they're the troublesome group that we've always stumbled over in terms of classifying risk. So if that's not bad enough, there's another dimension of confounding variables in risk assessment, which is epigenomics. And this is a very tough problem to study and a tough problem to understand. It's basically the fourth dimension. We have to learn how to deal with it now that we know that in AML and many other diseases, many of the mutations that are associated with these epigenetic signatures that have been confusing in the past are clearly related to mutations in genes that are epigenetic modifiers. So the biggest one for us has been DNMT3A. So this is one of the two cytosine methyltransferases, one of the de novo methyltransferases. There are 3A and 3B, which along with DNMT1, the maintenance transferase, is responsible for the methylation patterns we see in a genome. And so DNMT3A it's interesting, the patterns that we see. In AML, there's this one position, R882, which is about 60 to 65% of the mutations that we see fall in the single amino acid. The mutations that are seen there are almost always heterozygous, suggesting that they're probably acting as some kind of a gain-of-function property or a dominant negative. The rest of the mutations seem to generally cause loss of function, many truncating mutations, and frame shifts in the upper part of the gene. But one thing I just want to mention about this, you read a lot about modeling DNMT3A mutant AML with loss of function models. So it's very rare that this gene is deleted or haploinsufficient or not present at all, which is what the knockout models generally are doing. Most of these are point mutations that are having other effects, and most of them we don't really understand yet. And this is one of the things that David Tremaine has been studying in the lab. So the R882 mutation is really a special case. It's the most common mutation. It's actually in almost 20% of patients with normal karyotype AML. And it pretty clearly is the initiating event for that disease. It's very common, 10 or 15% of people with myelodysplastic syndromes. And it's probably the most common mutation in these elderly patients with skewed hematopoiesis. So this mutation is out there, and it's in many, many people. So how does it work? Well, one of the things David did in the lab was to show that it is a hypomorphic enzyme. Its activity, the mutant protein, is reduced by about 80%, and it's a very potent dominant negative for the wild-type enzyme. In fact, it reduces the total activity of DNMT3A in the cell by more than 80%, 80 to 90%. And importantly, DNMT3B, which is the other de novo methyltransferase, is basically absent in AML cells. It's expressed at reasonable levels sometimes, but it's almost all inactive splicing isoform of the protein. So the only active DNA methyltransferase other than DNMT1 in AML cells is this enzyme DNMT3A. So the question is, you have a mutation now that potently inhibits the activity of this enzyme in an AML cell. So what impact does it have on DNA methylation, on chromatin organization? It should do something, right? And it should have a gene expression signature. In fact, when we sent this original work in for publication, all the reviewers and the editors said that this will be a New England Journal paper when you understand what the target genes are. And I said, well, then probably this paper is never going to be published because we still don't know. After 5 years of intensive work, as I'm going to show you in a minute, we still do not have a handle on what this mutation is doing to specific gene expression that is meaningful. So rather than show you a bunch of heat maps of altered DNA methylation, I'm going to show you a much simpler version of DNA methylation throughout the genome. That's a simple XY plot. It's less biased than the kinds of heat maps we see of DNA methylation frequently in the literature, which are usually focused on differentially methylated regions, which are usually very, very tiny components of these genomes, which I'll show you in just a second. So what you can do with a simple XY plot is you can plot the methylation status of every gene in the genome, or every CPG in the genome, and you can have a control genome and an experimental genome, and if the CPG methylation values are unchanged, they're going to fall in the X equals Y line. If they're hypomethylated in the experimental sample, they'll fall over here, and if they're hypermethylated, they'll fall here. So what do we see in these AML genomes with and without the DNMT3A mutations? So this is David's work also. Here are AML genomes where we're comparing AMLs that don't have DNMT3A mutations down here to ones that have non-RAD2 mutations, and there's no methylation phenotype. Red dot means it's significantly different in the experimental sample. So these non-RAD2 mutated DNMT3A genomes don't have a methylation phenotype. So we got trouble already, right? And these RAD2 have a very, very clear methylation phenotype. And it's all over the place, but it only comprises 6% of all CPGs. They tend to be clustered together. They're canonical. They're the same, basically, in all AML genomes. So they're canonical. And virtually all the differentially methylated CPGs are hypomethylated. So it's a very direct connection to this mutation, which causes reduced activity of the enzyme. If you look, this is all array data. So we've gone on and done whole genome bisulfite sequencing from a number of these cases. And if you look at the whole genome at their methylation, again, just what I showed you, the RAD2 genome is here, and the wild-type DNMT3A genomes are here. These are very, very closely matched samples. There's just a little group of CPGs that are relatively unmethylated in the DNMT3A R882 cases. So it's a very subtle effect across the whole genome, but at some places it's really profound. So let me show you summary data from 8 cases of AML, 4 with an R882 mutation, and 4 that are matched in every characteristic we can think of, but they don't have an R882 mutation. So we'll do integrated epigenomics, and we're going to do it at just one locus, this NSD1 gene, which is a histone demethylase. So you can see the methylation phenotype up on top. So this is a canyon of cytosine methylation in both these wild-type DNMT3 cases and the mutant ones. There's this canyon here. So in every cell in the sample, no CPGs are being methylated in this canyon. This is 100% methylated, and this is 0. But you can see in this left canyon wall, you see how it's eroded in the mutant sample? So it's hypomethylated in that canyon wall. This is the most characteristic finding we see in these genomes in terms of the methylation signature. So we've got a little gray bar over this. This is Dave Spencer's work in the lab. So you can track this down as we look at all of the other components of epigenomics in this case. So here's a TAC-seq, which is a poor man's DNase I hypersensitivity, and it simply demonstrates regions of open chromatin. So this gene is relatively closed in the wild-type AMLs, and there's this beautiful region of a TAC-seq hypersensitivity, which is the DNase I hypersensitive site, that's only in the mutant genomes. So it's clearly altering chromatin structure at this region. Let's look at histone modifications here. So here's histone 4 trimethylation. So here are the wild types and here are the mutants. So there's a beautiful region activation mark right at this same hypomethylated canyon wall. And here's the K27 repressive mark. There's nothing there. And here's this, this marker for, I'm a globin guy. It's hard for me to say super enhancer. I have to say locus control region, all right? So here's a super enhancer mark, histone H3K4 monomethylation, right over the same hypomethylated region. So now you would expect there to be a big expression phenotype here, right? We certainly would expect that. It should be activated in this hypomethylated open chromatin super enhancer mark. Should be a huge change in expression. There is not, okay? Here's the RNA-seq data. Here's the top strand data for the wild type and the mutant. There is about a 2-fold difference in the read counts for this particular locus. Here's exon 1, here's exon 2. Very small change, not statistically significant because there's some variation among the cases. And on the other strand, there's nothing. And in the entire look at small RNAs from 17 to 200 nucleotides, there is nothing dysregulated in this locus. So a bit of a problem, and we're going to talk about this more. So the phenotype, though, when we've done lots of these whole genome bisulfite cases, the hypomethylation phenotype is almost always in these canyon walls. So here is the center a CPG island. And here are the data from CD34 cells and from wild-type DNMT3A genomes. And they all kind of fall to the bottoms of these canyons. And this outlier on the canyon walls, these are the R882 genomes. So this is 14,000 different loci in the genome. So it's a canonical signature. These canyon walls are where you get erosion and you get the R882 phenotype and methylation. And the other canonical feature of these things is that, again, centered on the middle of these differentially methylated regions, there is this open chromatin that's detected by the attack-seq or DNase-1 sensitivity in the same exact region. So there really is a perturbation in methylation that's in canonical regions of the genome, and there's a perturbation in chromatin structure at the same sites. Okay? So there should be changes in expression. And it should be very, very easy to detect them. But there is not. So here's a global view of RNA-seq data, very deep RNA-seq data, strand-specific RNA-seq, most modern techniques, really, really deep read counts for all of these cases. So 4 that are wild-type for DNMT3 and 4 of these R882 cases. So the expression levels for each annotated gene and all link RNAs are on this X equals Y plot. And there is just nothing here. There are hardly any genes that are dysregulated. We can do this also for small RNAs from 17 to 200 nucleotides, which we've examined in every one of these cases, and there is nothing that is obviously dysregulated. So you're saying, well, you should just be looking at the regions that have differentially methylated promoters. So, okay, here those are. X equals Y. So is this something weird about AML? So fortunately, we have a beautiful DNMT3A knockout mouse where you've completely eliminated activity of the enzyme. And you would think that if you take marrow from these mice, they should have the same hypomethylated phenotype, right? And they do. Here's an XY plot of overall methylation at all CPGs in these genomes, 3 on each axis. Beautiful hypomethylation phenotype, focal, canonical, a few percent of CPGs, just like the human AMLs. And here are all the differentially regulated genes in these DNMT3A genomes. By the way, the cells in the marrows of these mice after they're transplanted are not significantly altered in any way for at least 4 to 6 months. So the populations in these compartments are exactly the same. Same progenitors, same everything. There's just no phenotype. There are a couple of outliers that you're looking at here, but these are regions of the genome that are probably just mapping errors. So very, very few dysregulated RNAs. So this brings us to this huge conundrum, is that these methylation mutations are changing the methylation state in specific places in the genome, and changing chromatin structure are not obviously changing expression. So these R882s are doing something important to methylation and chromatin, almost nothing that we can measure at the level of RNA. So the question that quickly arises is, are you just missing something because you're comparing AML to AML? Is it that all AML genomes, no matter what they get started with and what perturbations happen along the way, they all end at the same place? So are they converging on the same phenotype? So when you compare these same AMLs to normal CD34 cells, you begin to get a sense that this may be happening. So here's another expression XY plot. We're comparing CD34s to AMLs, to these 8 AML cases. If it's red, it's dysregulated in R882, so there are a lot of red dots. If it's blue, it's dysregulated in wild-type genomes, and if it's purple, it's in both. So it turns out there are a lot of dysregulated genes compared with normal CD34 cells, or HSPCs, that are dysregulated in both compartments. And if you look at it on a heat map, here are the CD34s. So they have a different signature than the 8 AMLs. But the AMLs, regardless of whether they have an R882 mutation, have essentially the same signature. So the mutation status isn't governing this dysregulated phenotype. It's things that happen between initiation and the end state, which is what we're measuring. And everybody got to the same place. So I guess Yogi Berra's hypothesis of AML pathogenesis, when you get to the fork in the road, take it. So I think multiple paths may lead to the same state. I have no idea how this is really happening, what these rules might be. But the things that should have happened here didn't. And this is where we're sitting right now with this kind of data. So last thought I want to mention is that it's important to be thinking about how to use this kind of information going forward. Can we really be thinking about some of these very specific initiating mutations as therapeutic targets? And I think it's conceptually important to focus on these mutations because they're present in all the AML cells of a patient, right? If it's an initiating event, it stays around in all the subclones. And so the only mutations that are going to be targets in every subclone are the ones that were involved in initiation. So, our greatest success, of course, in AML is PML-RARA alpha, right? Which is clearly the initiating event for acute promyelocytic leukemia. When you put that fusion gene into a mouse, the disease phenocopies after it acquires cooperating mutations. But other mutations like TET2 and ASXL1, which clearly are initiating events for many patients, are also good targets to think about. So targeting mutations that are in subclones, as I mentioned earlier, like FLT3 or IDH1 and 2, can clearly produce beautiful clinical responses, but if they are in a subclonal place in the hierarchy, they're not likely to cure anyone. And if you think about it for a second, this is probably why allogeneic transplantation works, right? Because the alloantigens in AML cells are present in the founding clone and in all the clones. So I think the reason that the standard immunologic therapies work is because it's founding clone therapy. So what about this particular mutation as a therapeutic target? It's in a lot of people, so there's some interest in pharma in thinking about this. And one of the important things that Dave Germain did in the lab was he worked out the mechanism of this mutation, and it's pretty nifty, and it means it's targetable. So the idea is that DNMT3A wild-type molecules make dimers, homodimers, that make dimer-dimer pairs. So the active form of the enzyme is a processive homotetramer that rolls down DNA, finds CPGs, and decides whether or not to methylate them. Don't know those rules, but that's how the wild-type enzyme works. But the mutant, this R882 mutation, not others that we've tested so far, but this particular one is made in AML cells in stoichiometric amounts to the wild-type allele, always heterozygous. This mutant molecule has a huge advantage for binding to and locking down the wild-type protein. So this heterodimeric state is the one that basically eliminates DNMT3 activity in the cells. That's the dominant negative activity of R882H. So the idea is pretty simple. If you could somehow block this interaction, prevent it, the small molecule that was in the cell that allowed the wild-type dimers and tetramers to form, but prevented or broke the heterodimeric bonds, it could potentially restore wild-type DNMT3 activity, and you could think about it as a drug. So to that end, we've done two things. One is David made this very clever assay for a screen of small molecules that could break this interaction. So it's a FRET-type assay with these alpha lysa beads. So he made a wild-type DNMT3A full-length molecule with a V5 tag and a mutant one with a flag tag. When these molecules are juxtaposed and very, very close, when you shine light on them, the acceptor bead glows. So when they're together, you get a signal in a well. So this is easily adaptable to high-throughput screening technology. So this shows you that the wild-type interaction is much weaker than the wild-type mutant. This very, very tight interaction between the wild-type and the mutant is what we have to break. So we actually have completed a screen of a little over 500,000 compounds using this assay with Rigel Pharmaceuticals. And believe it or not, we got a bunch of hits that look to be fairly specific for breaking the wild-type mutant interaction. So we have to do some chemistry on these hits. It's way too early to know if this is going to work, but we're encouraged by what we see so far. And it then immediately raises another question. If you could restore activity to these cells, if you could restore DNMT3 activity, would it work? Would the cells remethylate? I don't know about expression, but would they remethylate, and would they remethylate correctly? So we did this experiment genetically. This was done by Angela Verdoni, a postdoc in the lab, and Shamika Ketkarn, a graduate student. We just asked a simple question. If you can add human DNMT3A in this case to a 3A null mouse, can you restore hypomethylation phenotype in those bone marrows? So Angela made these mice. It's an TET-inducible transgene with the wild-type human DNMT3A cDNA. And she bred that into the RTTA activator mouse, and we bred that into a 3A null mouse background. These mice die when they're 2.5 weeks old, so just harvest their marrow, transplant them into wild-type recipients, give them docs or not, and then do whole genome bisulfite of their marrow at different intervals afterwards. So here's what happens. So remethylation happens very quickly. Almost all the CPGs that are unmethylated because of the 3A loss are methylated by 2 weeks, and that continues to go up by week 9, and we've looked at it to week 24, and it's basically done sometime between week 9 and week 24. So you get remethylation. Let's look at one of these canyons. This is a canyon around the P2 promoter of a RUNX gene. Here is a wild-type mouse marrow, and here is a 3A mutant mouse marrow. Here's that canyon wall erosion I was telling you about that's characteristic of 3A loss. So here's the canyon, okay? Both kinds of animals have no methylation at all in the canyon, and then the canyon wall is eroded by the 3A loss on both sides. So watch what happens when we add back 3A 2 weeks later, so that'll be below here. So here's what happens at 2 weeks. This is what I just showed you. Here's the new data. So blue means no dox was added, no DNMT3A. Red means it's added. So what's amazing here is here's 2 weeks. So this is already recovered on the left side. It's getting there on the right side of the canyon. 4 weeks. This side is completely recovered. This side is getting there. 9 weeks. This side is hypermethylated. This transgene is overexpressed 4-fold compared to the endogenous allele. And on the other side of the canyon, it's completely restored. What is amazing, though, is in the canyon. See in the canyon? There's no methylation. So something is telling this enzyme, which should methylate all these CPGs in the canyon, not to do it. So we've looked throughout these canyons, and there's no abnormal methylation that occurs here, which I think is a very reassuring sign that you can have this remodeled genome, add back a wild-type enzyme, and have it go back to something resembling a normal state. This is one example of one canyon. So we've looked across many CPGs in these genomes. So here are a bunch of sites we selected because they're completely methylated in wild-type bone marrow, unmethylated in 3A. They don't get remethylated if you don't add DNMT3A, no dox. If you do add dox, some get remethylated right away, 2 weeks, like that one side of the canyon. Some are slower, but they recover later. And some are probably noise, probably things we misidentified, don't seem to get remethylated at all. But if you look at the canyons, there's no remethylation anywhere. So the canyons, which are not supposed to be methylated, stay unmethylated, and specificity is somehow preserved. So that's it. That's where we are right now. Coming back to this simple model. It looks very simple, but it actually has explained many of the clinical features of this disease. That's what I was getting at when I was explaining the clinical features. It's got to explain heterogeneity. It's got to explain the aged phenotype. It has to explain the canonical nature of the number of cases that are seen in the country every year. They're all a series of predictable events based on mutation frequencies, numbers of stem cells, how the first mutation occurs, how it modifies that cell to accept additional hits, and how it goes on to develop subclones that have unique properties. So I'll stop there, and I'll just acknowledge the many patients who have participated in this study. Our main funders, the NCI, NHGRI, and TCGA, Al Siteman, who funded the sequencing of our first genome, and the many people at the McDonald Genome Institute, especially Rick Wilson, Elaine Martis, Lee Ding, the Griffith brothers, Chris Miller, Allegra Petty, the main analysts that did the work I showed you today, and the people who have participated religiously in our Genomics of AML PPG, especially John DiPerzio, who leads our oncology group, and these postdocs and students who produced most of the data I showed you today. And this one here in particular is NIH property. So thanks all for your attention. I'll stop and take questions.
So questions, Lou?
So you made a convincing case that the DNMT3A doesn't really work through cis dysregulation. But you also said there sort of was convergent evolution of a phenotype.
Yeah.
So I'm wondering, this is sort of a weird hypothesis, but can all this massive change in chromatin be a sink for regulatory factors, chromatin modifying enzymes, that basically rob those things from a lot of other genes? So you could end up having sort of multiple loss of tumor suppressors, maybe in a haplo-insufficient way, that then leads to more proliferation. And if you thought that, if it's multiple, I don't know how I would do an experiment to get at it, but if there was one that was particularly important, then you could imagine a CRISPR-based method for finding that if you had the right phenotype to score.
Right. You know, I don't know what the answer is to this problem. I mean, your hypothesis is as good as any. We measure the things we can measure, right? I'm not saying that we have ruled out every possible, thing that could be changed by these altered-looking chromatin states and methylation profiles. Just because they don't alter gene expression in cis doesn't mean they're not doing something important. It could be nuclear matrix.
Exactly.
It could. There's so many levels of things that we're not measuring that I'm hesitant to support any particular hypothesis, just that everything ends up at the same place at expression, which is a really curious thing, maybe not terribly surprising.
Well, I mean, that I had a little bit of quibble with, because some of that is just proliferation.
Sure.
So,
yeah,
so
possible.
that's not maybe a
I'll
strong
give you
enough,
that.
a carefully enough narrow phenotype to talk about, but somewhere in there something gave rise to that proliferation.
Yeah.
That might be the phenotype.
This is what you get with statistics, right?
Let's go over to this microphone.
In pathology, I remember a long time ago we saw cases with what was called a granulocytic sarcoma or a chloroma, which was considered to be a prodrome to AML. And they were often recognized as, we thought they were malignant lymphomas, etc. Now, I actually had done a presentation of a case, and 40 case studies were done at Roswell Park when I was there. And it was, my understanding is that they're actually a prodrome for AML, a chronic granulocytic sarcoma or a chloroma. Do they have any significance in terms of their genetics, their overall genetics, or at what point a chronic smoldering disease pops a chloroma, or as we used to say, granulocytic sarcoma, and becomes AML? In other words, is that a critical tipping point in terms of genetic mutations? I think that's valid because in the literature it's seen as a prodrome to AML.
Yeah. I think now most people would consider it to be AML. It's extramedullary AML. And the other kind of interesting thing that we've learned about extramedullary sarcomas over the years is that quite frequently after allogeneic transplantation, this is the site of relapse. It's extramedullary. I can't say it. We haven't really sequenced any extramedullary AMLs that have presented extramedullary, but we have sequenced now a number of extramedullary relapses post-allogeneic transplant. And they're derived from the original founding clone, so there's something that's changed about them that makes them more likely to sit down in an extramedullary site. So they're not relying on the medullary niche, if you will, the stromal cells in the marrow for their growth and differentiation, which isn't surprising. It is kind of interesting why this post-allo relapse is extramedullary frequently and post-chemo isn't. My guess is it's just a form of AML that has slightly different mutational profiles. I don't know what the answer is because we haven't sequenced any of these cases, but it's a pretty bad disease, extramedullary AML, as bad as AML.
Thank you.
Yeah. Ron.
So let me take a different version of what Lou asked you about.
Yeah.
All of these experiments are still on bulk material, and you're not looking at what the individual cell state is. And so if there is a phenotype in which occasionally these eroded shoulders give you transcription for a short period of time that then drives something in the cell, you won't pick it up this way. And so I wonder, I know, you guys have been trying to get down to smaller and smaller numbers.
Yeah.
It's still not single cell work, and it'd be tough to do this, especially if you don't know what you're looking for and how many cells to look at. But it really raises this question. Given everything on the molecular side, it's very hard to understand how there could be no effect on transcription. But if there's a limiting factor, and this is what relates to Lou's part, that has to be expressed at that moment in the cell to target that site to get the transcription to go, then you're going to get this very episodic behavior, and that may be enough to drive the cells and always keep them in a stem cell-like state, but you don't see it this way. So are you moving to try to re-explore this question at a finer grain level is really the question.
Yeah. It's the problem with all epigenomic measurements. They're made in bulk, and therefore they're averaging technologies, unless you're doing single-cell work. So none of these have been adapted to the kinds of levels that where we could even think about doing single-cell stuff. I understand your point completely. The one thing that helps us a little bit is the AMLs are clonal, right? So in AML, even without purification, the vast majority of the cells we study in one of these unfractionated marrows is from the founding clone. And when you look at varying allele frequencies of all these mutations all these cases I showed you, the DNMT3A mutation is in every cell. That helps, but doesn't answer your question directly. It's helpful because at least you know every cell that you're measuring has a mutation, and that you're probably not looking at some averaging, in these cells. They're truly clonal populations. In that mouse marrow stuff, that's a problem because that's whole marrow. But still, think about it for a second. When you see something go down to ground in unfractionated mouse marrow, a methylation phenotype, there's only one way that can happen. It's in every cell in the sample. So it tells me that a lot of these methylation marks are also being added at the stem cell level because they're being transmitted with fidelity to every lineage and every cell in the sample. That's the thing we can't measure, time with these assays. And it's a conundrum that we haven't solved yet, mainly because we can't study these cells over time, and we can't study very, very small numbers of cells because of the nature of the technology. So point granted, I think it's a very valid point and one thing that we need to get to. The studies we did were the studies we had to do. We had to start. It made sense to start there. The conundrum comes when you get the data and you go, do we miss something? It's not obvious what the hell the answer is.
Hey, Tim, Marston Linehan, thanks for the really wonderful talk. I think it's one of the most beautiful stories in all of cancer genetics. And as I've told you before, those of us working in solid tumors are really hoping to go to school on what you've taught us, what you've learned. Let me ask you a quick question.
You're doing all right.
Well,
yeah.
I don't know. Well, we'll talk in tangent. Let me ask you a question. Let's say you have a tumor, let's say you have cells that has either DNMT3A or TAT2 mutation, okay? All the cells. And you also have either FLT3, IDH1 or 2, many of the cells. Is it your perception that to get home with an effective form of therapy, you're going to have to get both of them, or just go after the, let's say, truncal mutation, the DNMT3A or TAT2?
That's my opinion, is that going after the mutation, if you could really hit the initiating events, even without knowing what they do, exactly, Lou's hypothesis is a good one. Who knows what the hell these things really do? At least you know it's doing something to the epigenome. And I think that since you know it's in every cell, and therefore in every subclone, that I think it's the best target, to try to target,
Short question. I'm interested in the RNA data. When you look down the DNMT3A methylation activity, you say RNA unchanged. I wonder what do you mean RNA? microRNA or
everything?
Everything.
We've sequenced all of those compartments in detail.
In my
microRNAs, SNOs, CD boxes, you name it. All annotated and unannotated RNAs from between 17 and 200 nucleotides have been sequenced from all these.
In other cellular models, so especially in my impressions,
it's as good as we can do.
3A, 3B is very sensitive to cellular states, especially the embryonic states in development. And they are very sensitive to change in microRNA. So that is some notes I'm sure so probably is that be sensitive in our model. Thank you.
We definitely have more work to do.
Okay,
Thank you.
well, thank you.
You bet.
Thank you. Marshall would be pleased. And let me invite you all to an FAS-sponsored reception in the library where you can continue some of these discussions. Thanks.